Thank you. The next speaker of the session is joining us online. We have with us Professor Neil Burgess, University College London, UK. He will be speaking on the topic, neural mechanism of spatial and episodic memory. Professor Neil Burgess is currently Professor of Cognitive and Computational Neuroscience at University College London and Wellcome Trust Principal Research Fellow. He was awarded a PhD in theoretical physics by University of Manchester in 1991. In Manchester, he began modeling working memory with Graham Hitch. He returned to UCL to work with John O'Keefe, working on models and experiments concerning how neurons represent space and support memory. His behavioral neuroimaging and electrophysiology experiments with both human and rodent bridge the gap between brain science and observed human behavior, shining a light on the neural representations and computations supporting spatial cognition. With colleagues Tom Hartley and Colin Liver, he predicted and discovered neurons representing environmental boundaries. With Sue Becker in 2001, he proposed the first model explaining how neurons in the hippocampal system support coherent spatial imagery. This model has developed to explain aspects of episodic memory, imagery, and planning in terms of the activity of populations of neurons. He has a rich research experience of more than 4 decades with over 200 publications in journals of international repute and well-cited with H index 96. He also held many responsible positions in academia. For his work, he has been recognized with many awards, to name of very few, William James Fellow Award for Lifetime Contribution to Psychology by Association for Psychological Science, elected Fellow of the Royal Society, and elected member of the International Neuropsychological Congress. Please help me welcome Professor Neil Burgess.
Right, I shall share screen. Great. Well, thank you for inviting me to talk at this very interesting summer school, and I enjoyed the last bit of the previous talk. What I'm going to try to do today is say something about what the neurons are doing when we remember things.
Can you hold on a second? We
Okay.
are trying to connect your mic.
Right.
Can we check it once?
Hello, can you hear me? Can you hear me?
Yeah, now?
Yeah, can you hear me now? Can you hear me? 1, 2, 3. Let me know when you want me to say something.
All right. Yes, sir.
Can you hear me now?
Yes, we can continue.
Great. Okay. Thank you very much. So I'm going to try and explain something about what the neurons in our brains are doing when we remember things. And please interrupt if there's any technical problems. First of all, I'm going to talk about neural representations based on spatial memory representations that we've understood quite a lot about in the last 30 to 40 years. And then I'll slowly move on to a more general topic of how episodic and semantic memory can function together in our brains. So to start with, we know that the hippocampus, as we heard in the last talk, but also from your general textbooks, the hippocampus is required for episodic memory, the ability to remember what happened yesterday or in the past, and to sort of vividly re-experience what happened as you think about it. And we know that since the famous patient HM, who became amnesic after his hippocampi were removed to cure severe epilepsy. And as I said, what I want to know is what the neurons are doing. And if we look at spatial memory, then studies in rodents have been very informative, where here's the hippocampus of a mouse or rat. And if we implant electrodes chronically in the hippocampus to record from neurons there, while the rodent is behaving various tasks or performing naturally, then starting with this discovery from John O'Keefe of place cells, we can see what these neurons are doing. And so here's an illustration of a rat exploring in a box and the recording from one of these neurons in the hippocampus. And a little red dot is shown each time it fires an action potential. I'm not quite sure how well this comes across, but what you'll see is that as the rat explores, looking for randomly scattered bits of food, then whenever it comes over into this place here, that single neuron will fire an action potential. And this is representing the place where the rat is. And that's why O'Keefe called it a place cell. What I want you to notice is that it doesn't matter which way the rat is facing. It could be going in any direction through this place, and the cell still fires an action potential. And this immediately tells us that it's not a simple sensory response, because a sensory response, particularly a visual response or an auditory response, would depend to some extent on which way we're facing. What we can see when we're facing one way is different to what we can see when we're facing the other way. And so this kind of response to location or representation of location was called allocentric. It doesn't depend on the orientation of the body. It indicates where you are in the world. And since the discovery of these place cells, this shows the track the rat took, and these red dots again show where the rat was when a given single place cell fired an action potential. There were also discovered head direction cells. And these are complementary to place cells. Place cells fire when the rat's in this place and don't care which direction it's facing. The head direction cells fire wherever the rat is, but only fire when it's facing or heading in a particular direction. So one place cell will fire here. Different place cells in the hippocampus will fire when the rat's in different locations, and together they're telling the rest of the brain where the animal is. The same is true for head direction cells. This cell here fires when the rat's facing in one particular direction, and different head direction cells will fire when it's facing in different directions, and together they will tell the rest of the brain which way it's facing. More recently discovered grid cells, shown here, we'll come back to a little bit later. For the place cells, if we record one place cell shown here, so it fires when the rat goes into this location, what we can see is if we make the box that the rat's exploring in, if we stretch it in size by moving the walls of the box relative to each other, we can see that this single location where the cell fires gets stretched out, and what you can see is that the cell is actually getting inputs which tell it that the rat's in the right place for it to fire, because there's a wall quite close to the north and quite a little bit further away to the west, and maybe even further away to the east. And those separate inputs to the place cell can be seen in these larger boxes. They all converge in this location where the cell fires with its typical unimodal firing field. So we propose that actually what tells the place cell where the rat is to make it fire are what we call boundary vector inputs. So they're inputs that tell the rat when there's a boundary at a given distance and direction away from the rat. Since then, we found these so-called boundary vector cells around the hippocampus in the subiculum and entorhinal cortex, which is the cortical area that inputs into the hippocampus. And each of these columns shows a different example of a boundary vector cell recorded in different environments. So these are two recordings in a square box with a curtain around. This is a circular box with no curtain. This is actually the tabletop on which the box, the square box, was put. And what you can see is that this cell, this boundary vector cell, fires whenever there's a boundary a short distance to the east. It doesn't matter which box the animal is in, or even if it's on this tabletop and the boundary now is the drop at the edge of the box rather than a drop at the edge of the table rather than the walls of the box. So these cells seem to really just care where are boundaries around the animal. Here's some other examples showing what happens if you put a wall, a little piece of wood inside the box. This cell fires whenever there's a boundary a short distance to the south, whether it's the original boundary of the box that it's in or this piece of wood that you've just put in. Or here's an example with some bottles of wine have been put into the box. So these cells, they don't care what the boundary is, they just care where it is relative to the animal. And since then, object vector cells have been discovered in entorhinal cortex by the Moser group. And here's 3 different examples of an object vector cell. These cells fire in response to discrete objects that have been put inside the box where the rat or mouse is exploring for randomly scattered bits of food. And again, they fire when there's an object at a particular vector away from the animal. And again, it's an allocentric vector. This cell fires whenever there's an object short distance to the east of the animal. It doesn't matter which way it's facing. That neuron will just fire whenever there's an object to its east. And it doesn't matter what the object is. So this might be one object and this might be another object. The cell fires whenever there's an object a short distance to the east. And here's another couple of examples with more objects and different directional tuning. So these are all allocentric responses that indicate where the animal is in an environment, which way it's facing, and perhaps the presence of objects and boundaries around it. But we mustn't forget that most of the brain is full of egocentric responses. So visual cortex, sensory cortices are usually full of neurons that respond to particular stimuli in a way that depends on where it is relative to the animal, whether it's on the left or the right, and therefore depending on which way the animal is facing. So a typical receptive field for a visual response in visual cortex would be if the animal is looking in this direction, then a stimulus maybe down here relative to where the animal's looking will elicit the maximum response. And if the stimulus comes somewhere else, then it won't make the cell fire. So in sensory cortices, there are lots of egocentric responses which respond to what's around the animal, but depend on which way the animal's facing. And therefore, for example, visual responses depend on where you're looking and where the stimulus is relative to where you're looking. Similarly, output, motor cortex is full of neurons that are also egocentrically tuned. Different neurons will fire according to whether you need to turn left or turn right or reach in a particular direction relative to your body. So if we want to understand how the whole of the memory system focused on the hippocampus works, we need to take into account the fact that sensory information comes in egocentric representations, and then the hippocampus seems to be full of allocentric representations, for example, about where you are but not which way you're facing, or about which way you're facing but not where you are in the world. And then output from memory, either the decision to turn left or turn right, or maybe to imagine, as you remember what happened, imagine what was on the left or what was on the right or what was straight ahead, so that you can actually imagine a coherent scene. That needs to be an egocentric representation again, because you need to know what is left and what is right and what's straight ahead. And if you were to turn around, then that would change. So if we want to try and make a simple model of memory and imagery for simple scenes, for example, of what happened when you had lunch with your friends yesterday, we need to understand how these egocentric sensory representations and allocentric representations of space in the hippocampus and around the hippocampus, how they interact with each other. And a nice solution originally from Puget and Sinowski and recorded in experiments in primates is that there are also gain field neurons. So these are neurons that respond to, for example, when a stimulus is on the right or on the left, but also multiplied, gain modulated by which way you're facing. And if we think of a simple circuit like this, where there's some neurons which encode whether there's a stimulus on your left or ahead or right. So maybe here's the rat and it's facing north, and there's something here to its right, then maybe this cell would be firing if this is a egocentric object vector cell. And these might be the allocentric object vector cells. There's a neuron that fires whenever there's an object to the east of the animal here. If we want to map that right means east, we need to know which way we're facing. So the fact we're facing north, and maybe there's a head direction cell that is tuned to fire when the rat's facing north. If we know that we're facing north, then we know that what's on our right is to our east. And if we were to rotate, then if we're facing east, then the object is still to our east, but we know that it'll be more close to straight ahead. And so if we have neurons here, which you could think of as a copy of these neurons, but a separate copy for when we're facing north or east or south or west, then which one of these neurons in the middle will change? And these neurons here in this population, which are gain field neurons with responses to where stimuli are relative to us, but also firing when we're facing in a particular direction, these can map the activity from this neuron through to this neuron, whereas this neuron can map the activity from this one to this one. And which neuron maps the activity correctly depends on which way you're facing. And you can easily make a simple circuit like this with a simple learning rule that will connect all of the appropriate neurons together according to when they fire simultaneously. In fact, this whole circuit has recently been found in flies. If you look at the fact that there are neurons in the fly that encode the fly's heading direction relative to the external world, there are also neurons in flies which fire when the fly is going to move forward or left or right. And there are also, they recently found neurons which fire according to whether the animal is going to walk north or south or east or west. And the way they're all connected is via these gain field neurons in exactly this way. And you can even see this pattern of wiring up in this gain field neurons that maps the activity from left, right, straight and ahead to north, south, east and west. So with Sue Becker in 2001, as we heard in the kind introduction, and more recently with Andrzej Bichanski, we showed that you can make a computer simulation of place cells and object vector cells and boundary vector cells and cells encoding the identities of the objects and boundaries, so that connections can form between all of these different kinds of neurons as an animal explores an environment. And that will encode that environment so that it's a familiar environment, and the rat or mouse, if it goes back there, or we think person, as we'll come on to later, will know if they're in a particular location, they'll know what objects and boundaries to expect around it. And during learning, egocentric sensory information has to come in and be translated via these gain modulated neurons. We think this happens in the retrosplenial cortex, informed by head direction cells, so that they can translate these egocentric representations into allocentric representations, which can then go into this long-term memory network. And then for output, if you remember something, you can recreate where the objects and boundaries around you would have been in allocentric representations, so north and south and east and west of each other. And if you know your head direction, then you can translate that information back into egocentric representations and actually visualize a coherent scene of what would be on your left or straight ahead or right if you're facing in a particular direction. And so this would be a sort of comprehensive model of encoding into memory in this direction and retrieval from memory in this direction. And we can see here the importance of the place cells in providing a viewpoint from which to collect this long-term knowledge that's stored in and around the hippocampus, and project it into a visual scene from a particular head direction, particular viewing direction indicated by your head direction cells, so that you can create an image, a coherent image, which has a viewpoint and a direction. So if we do fMRI experiments with people, if we put them in the scanner and ask them to remember and imagine things that have happened in their past, then you see typical patterns of activity in the brain, including the hippocampus and the parahippocampus, retrosplenial cortex, and this medial parietal area, the precuneus. And with our model in mind, we can have some idea for what the neurons behind these increased metabolic activity that you see in an fMRI scanner, what they might be doing as you're remembering what happened. And so we can actually get some beginnings of an idea of what functions the neurons in these different areas are doing. So as I said, these boundary vector cells were predicted as the inputs driving place cell firing. And this kind of model, the whole model, also predicts that there should be egocentric versions of object vector cells and boundary vector cells, as well as head direction modulated versions of these cells for this egocentric to allocentric translation process. And nearly all of these different kinds of cells have now been found in various parts of the brain. So our network that we think of as a memory network, actually, it's more than that, because it's a generative network that is capable of generating coherent images, given your long-term knowledge of the layout of a particular environment, and the ability to impose a viewing location and a viewing direction. And this is also, we think, in humans at least, related to planning, imagining what's going to happen in the future. Psychologists have given it various names. But the point is that a generative network for constructing coherent scenes, it seems to be what episodic memory requires, so that you can imagine re-experiencing an event. But it's also required for various things like planning and imagination. So this model tells us something about what the neurons might be doing in the brain, and we've speculated about where these populations of neurons might be, but there's evidence from neuropsychology which helps us here. So an old experiment by Biziak and Luzzati, they took patients with hemispatial neglect, so that they're patients who've had damage to somewhere the right side of the brain in the parietal and prefrontal cortices. And typically they neglect to pay attention to things on the left-hand side. So they might just eat food from the right-hand side of the plate, or they might just comb their hair on the right-hand side. These patients were interesting because Biziak and Luzzati asked them to imagine scenes from their hometown in Milan. And they were very familiar, before they got the brain damage, with the Piazza del Duomo, the town square in the center of Milan, where there's a big cathedral. And they asked them to imagine being in the Piazza del Duomo, facing towards the cathedral, and to describe what they could imagine. And when facing towards the cathedral, they described these buildings here marked in green, predominantly on the right-hand side of that viewpoint that they were asked to imagine. But when they were then asked to imagine standing facing away from the cathedral and to describe that scene, they described these buildings marked in red, which are all on the right-hand side of that viewpoint. So what this seems to imply is that they formed an allocentric representation of the town square. They know the layout of the buildings relative to each other. But when they have to generate a imagery and create a scene that they can imagine, then because of their hemispatial neglect, they neglect what's on the left-hand side of that generated scene. And so the medial temporal lobe, the hippocampus and surrounding structures, is probably fine, and their long-term memory for this, this place is okay. But when they have to generate a scene to imagine, then their right-sided parietal damage is probably making it hard for them to pay attention to the left-hand side of that scene. And this is consistent again with the overall layout of that model. So another thing I'd like to mention quite briefly is that I've talked about so far how you know where you are given environmental information. You can look around you and see boundaries and objects around you, and that can tell you where you are and the place cells can fire. But you also know where you are because of how you've moved. So if I've taken an outbound path, this curvy path here, then actually because of how I know how I've moved, that tells me where I am relative to the start of that path. This is often called path integration. And grid cells in entorhinal cortex, just next to the hippocampus, found by the Moses in 2005, they are thought to be one of the ways in which the brain updates our representation of where we are, given how we've moved recently, or how this function path integration occurs. So here's a similar little cartoon of a rat exploring around, somewhat sped up in a large box. And one grid cell is recorded here. And you can see whenever it fires a spike, there's a little click and a red dot is put down in the location that the rat is when the cell fired a spike. If you collect plenty of data, you can see that this cell, this grid cell, likes to fire whenever the rat goes into any one of a number of locations. These locations are all arranged at the vertices of a regular triangular grid. And if you record from near the top of the brain, then you see small scale grids like this one, and further down you see larger scale grids. And if you record from many of these grid cells, particularly here, 3 grid cells with the same orientation and scale of grid-like firing pattern, you can see why people think that they might be a useful neural substrate for calculating path integration. And that's because they each have the relative locations of their firing fields are fixed relative to each other. So if I'm here where this red node is, the red grid cell will be firing. But if I move north, then the green grid cell will be firing, and that will be true wherever I am in this environment. It'll also be true across environments. Whereas place cells, different place cells fire in different environments, these grid cells fire similarly across all environments. And so it'd be quite easy to wire up for the activity to pass from the red grid cell to the green grid cell, for example, whenever I'm moving north. And that would be true wherever I start from one of these red locations. So the sort of hypothesis that most people are working on is that the place cells will get environmental input, like from boundary vector cells, that tell the place cell where to fire when the rat's in a particular location in an environment. And the grid cells update which grid cells are active according to the movements of the animal. But it's also true that to estimate where you are, your self-location, you should combine both of these kinds of information, the environmental information and the self-motion information. And indeed, the grid cells in entorhinal cortex project into the hippocampus where the place cells are, and the place cells project back to the entorhinal cortex where the grid cells are. So what's probably happening is that if a place cell fires in a particular place like this in the environment given environmental inputs, it will form strong connections with a grid cell that happens to fire in the same place, and vice versa. And that means that the place cell can know to fire in this location, even if, for example, somebody switches off the lights and there's some restriction or uncertainty in the environmental inputs. And equally, the place cell knows to fire here from the environmental information that comes through the place cell, because a grid cell couldn't keep integrating its own self-motion signal to know which grid cells should fire over many hours and days. For example, if you take a rat and you record a grid cell and you put it back in its cage and come the next day, the grid cell will fire in the same locations again. And that would be impossible through updating via self-motion. But by forming connections with place cells, then the grid cell can know, its firing pattern can be made stable relative to the environment. And indeed, if you, to test formally test this idea, we made a two-dimensional virtual reality system for mice where you can record the firing patterns of grid cells and place cells and head direction cells and so on. And in virtual reality, you can then change the gain between the visual information and the movement of the mouse running on a styrofoam ball, so that the self-motion information and the visual environmental information can tell you different things at the same time. And when you do that, you see indeed that the grid firing patterns more follow the self-movement of the mouse running on a styrofoam ball, and the place cells are more driven by the visual information from the environment. So coming back to humans, we wondered whether we could see these grid-like firing patterns in humans. And with Christian Dohler and Caswell Barry here, we realized that because grid cells, the different grid cells with different grid-like firing patterns, even the ones that have slightly different scale grid-like firing patterns, they tend to have the same orientations of the grid. So, for example, these orange directions here along the grid axes, they're the same, those 6 directions, for the majority of the grid cells in entorhinal cortex. And so the neural firing dynamics, when you're running along these orange directions, in which a given grid cell will either be firing a lot because you're running through all of the firing peaks in close succession, or firing very little because you're missing all of its peaks. That situation will be slightly different when you're running misaligned to these grid axes, shown in gray here. In these directions, most of the grid cells are firing, but less often, because you go through one firing peak and then another firing peak further away. This difference in the neural dynamics of firing, given how nonlinear the bold signal is that you measure in fMRI, the fact that we've got either some grid cells firing a lot or many grid cells firing less often, should give us a different pattern in the fMRI signal according to which way we're running, whether we're running aligned to these grid axes or misaligned. And if you look for that pattern of activity in the brain, while people are performing a virtual reality navigation task where they have to remember objects, locations, and explore in a circular arena, you can see an overhead view here, and this is the path of one person. While they're doing this memory task, we can look at the fMRI activity. And what we find in some parts of the brain is that the activity does indeed vary according to whether you're running aligned or misaligned, or aligned or misaligned to a grid that is has 6 peaks as a function of running direction. So every 60 degrees, you have a increase or a decrease in firing. And we see that in the entorhinal cortex on the right, and also in other areas like medial prefrontal cortex that are associated with autobiographical memory. So perhaps the grid cells in rats that we think update the representation of where the rat is by its own movements, maybe in the human memory system, they can enable you to update this viewpoint from which you can generate imagery to imagine what happens. And we tested that in an experiment with Aidan Horner here, where in the same virtual reality spatial memory task, when they were shown an object that they'd found somewhere in one of these virtual environments, they had to go back to where it had been and press a button. But before they did that, they closed their eyes and imagined going in that direction. And we saw the same grid-like signal in entorhinal cortex for when this imagined direction goes through either aligned or misaligned with what might be the grid cells in their brain, that we saw with actual navigation. And so it seems like they're likely that in humans, grid cells do update where we imagine we're located, and we can do that updating based on imagined location, if imagined movement, if we need to. So in this, okay, if we go back to this model, then we might need to add grid cells here to be able to change the place cells that indicate which location we're imagining being in, from which we can generate an image and imagine the scene. So in the last bit of the talk, I would like to say something a bit more general about human memory and not just spatial memory. So if we record from neurons in the hippocampi of, again, epilepsy patients like HM, but nowadays neurosurgeons are careful to remove the only one side of the hippocampus so that they don't become amnesic. But in the process of working out where the epilepsy is coming from, if it's drug resistant, then electrophysiological recording might be necessary. And Itzhak Fried's group showed a while ago that single neurons in the human hippocampus will respond to concepts such as Jennifer Aniston, famous actress, but a given neuron will fire to a picture of Jennifer Aniston, or even her name written down, for example. And also to pictures of famous places like the Taj Mahal or the Eiffel Tower. And in rodents, we see there are responses in tasks which require paying attention to different kinds of sensory stimuli, like the pitch of a sound. We get neurons that respond like place cells, but to whatever that important sensory stimulus was. And finally, in a beautiful experiment by Tim Barron's group, Alexander Constantinescu, trained people to become experts in bird silhouettes. So they watched the silhouette of a bird with the neck or legs getting longer or shorter, and they had to become experts in spotting when it was, say, a duck with both of them short, or a crane with both of them long. And having trained for a long time to become experts in these bird silhouettes, in the fMRI scanner, they just watched these silhouettes changing shape and size, and they had to know which the trajectory of these changes in the two-dimensional space of neck length and leg length. They had to pay attention to that to know if it was going to become a duck or a crane or something else. But just watching these silhouettes changing shape and size, and then doing the same analysis that we did in terms of the direction of this trajectory in this conceptual space, again, you see this grid-like signal in entorhinal cortex and medial prefrontal cortex. So perhaps if we go back to this sort of simple circuit understanding that we had from spatial memory, perhaps place cells in general in human hippocampus represent states or concepts, and grid cells represent the relational information or the transition information between these states or concepts. And we can then try to, understand human memory more generally. What I'm going to do right at the end now is to give a brief idea of how we might come up with a more general model of episodic and semantic information, using this idea that the hippocampus and neocortical areas actually represent a generative model, and relate this to how memory construction and imagination happens, and also how it's affected by consolidation. So from the original study of patient HM, it was noticed that the older memories that he had at the time of the surgery were spared. Although he became amnesic and couldn't remember new things, he could remember some things from the past, particularly his childhood. And this is another patient who kept an autobiography, PZ, so he could be asked questions about events from his past, which they took from his autobiography, going many decades back from when he had Korsakoff syndrome, in fact, which caused hippocampal damage in the 1970s. And here you can see this temporal gradient of retrograde amnesia, the ability to remember things, older memories from before the hippocampus was damaged. And rather like in the talk that you just heard, memories can become independent of the hippocampus, even though they require the hippocampus to be encoded in the first place. So this gave rise to the idea of complementary learning systems, or a model of systems consolidation, the idea that the hippocampus can rapidly capture what's happening to you as it happens, and store that for some amount of time. But over time, this information in the hippocampus can be transmitted or used to train networks in the neocortex, which can then eventually learn information or store information in a way that's independent of the hippocampus. But this traditional model has some challenges, and I'm going to briefly outline maybe an updated version of this traditional complementary learning systems model. So this is the model that initially the hippocampus is required for encoding, and then some transfer or training of information means that the neocortex can be trained. And this is how semantic memory is developed, which is extracted from lots and lots of different experiences. We extract the common statistical structure of what normally happens over lots and lots of events. But there are challenges. For example, Nadel and Moskovich pointed out that truly episodic recall may still depend on the hippocampus. So patients who can remember memories from long ago, who have hippocampal damage now, when they recall those memories, they seem to be in semanticized form. They know facts about what happened, but perhaps they're not able to fully experience or reimagine what happened in the way that, for example, Endel Tulving defined as the hallmark of episodic memory, to be able to re-experience. And they suggested that information maybe is from the hippocampus is used to train the neocortex, but actual episodic recollection might remain dependent on the hippocampus. And each time you think about something, maybe you lay down another trace in the hippocampus. And that's why old memories are more resistant to hippocampal damage than more recent memories. But the real challenge, I think, comes from the fact that memory is reconstructive. It's not veridical. So when you remember something, you can imagine a plausible scenario, and many details of what has happened will be correct, but actually some will be incorrect. And what you will have done is construct something that is likely to have happened and consistent with what you know. And indeed, some little details might actually have happened, but other aspects might not have. And so psychologists have known for a long time that memory is reconstructive. And more recently, if you put people in the brain scan and you ask them to remember stuff or to imagine new events which have never happened, it's often called episodic future thinking, then you see similar networks of activity in the brain for both of those activities. And it's also, going back to this semanticization, it's also true that as consolidation happens, as memories get older, then this can promote inference and generalization. So our semantic memory is very useful, but we also get increasing gist-based distortion. So we tend to remember things as more typical than they were. In the last few minutes, I just want to give you a flavor of an update to this systems consolidation, complementary learning systems model in terms of generative networks. So in artificial intelligence and machine learning, there's been a lot of progress recently in various machine learning methods for dealing with information, large amounts of information. And some of this corresponds to generative networks. And so here's an autoencoder is a kind of network where you just train it to get lots of input patterns and to reproduce whatever the input pattern is at its output. And in the middle of this deep network, there'll be a layer of neurons with not many neurons in it. So there's a bottleneck. So to be able to correctly reproduce a large number of different images from its input to its output, this neurons in this bottleneck have to form a very efficient compressed representation of the information that's in these images so that they can recreate lots and lots of different detailed images using only a small number of neurons. Particular kind of autoencoder called a variational autoencoder enables you to sample or give your own values to these neurons in the middle here and generate realistic stimuli that could be new ones, but they'll still fit with everything you know about the data set you've been trained on. So for example, if you train on a large data set of faces, it will, in an unsupervised way, learn to represent the the main features of information in the faces, such as their age or their gender or how much hair they have. And these representations in this hidden layer can then be used to generate, for example, if you have a starting face, like either of these, you could change its gender or change its age just by changing this representational in the center of the network, because it's captured these different dimensions along which characterize each image. So I'm not going to go into much detail, but if we go back to the idea of the hippocampus rapidly capturing information and then training the neocortex, like in complementary learning systems models, if the neocortex is actually a generative network, then we can see these smaller regions near to the hippocampus, like entorhinal cortex, could be forming latent variable representations, like these representations that are formed in the middle of a generative network that capture the essence of what's in all of the experience, which is being experienced here in sensory neocortex, just by having to reproduce that same input as an output. So the hippocampus can store things rapidly by forming associations between neurons. I won't go into any more detail about that. And this variational autoencoder here, it has a small set of neurons in the middle, and then it recapitulates its inputs as its outputs. But it's important to remember that the sensory neocortex here is the input and actually the output, because it has to recapitulate this imagined scenes, for example, or people in the sensory neocortical areas that the input came from. So the actual layout of this model will have sensory neocortex, various hidden layers of processing neurons, and then these latent variable representations near to the hippocampus, for example, in entorhinal cortex. And if you train this network on lots of simple scenes, what you can show is that the hippocampal network can rapidly encode the details of the scenes it's being trained on. And then by being given random inputs, the hippocampus will recall any of these scenes, which can then be used offline to train the neocortical model, because these generative networks need many, many examples of inputs and outputs to be trained on. And then once as the generative network gets trained, then it can reproduce even noisy versions of these input patterns as outputs, because it now knows what the structure of these scenes that it's experienced is. And this kind of model can explain lots of phenomena in the psychology of memory. For example, semantic memory, the latent variables here capture the facts in each of these scenes. For example, that this is a pink cube in the middle, and that information can be simply taken out from that hidden layer after training. Also, these networks show gist-based distortions. So for example, one thing we see is that is something called boundary extension, where if we see a scene, we recall it as more typical than we've seen it, so with the normal sort of layout. But I'm going to rapidly conclude now, if my computer will let me do that. And make some conclusions so we can have some discussion. As with generative networks, we can generate new scenes. Having been trained on some scenes, we can generate arbitrary new combinations of scenes, or even do inference of a complicated nature. And so this corresponds to episodic future thinking. And the details of this model we'll skip, but hopefully my computer will let me get to the conclusions, where it's been slowed down because of the Zoom. Where what we need, the hippocampus has to capture the unexplained unexpected sensory features that the generative model can't explain itself. And then the generative model can give the semantic support to generate scenes and images and recall memories without having to store every detail in the hippocampus. And this is an efficient way of doing things, which we think is how memory is organized in the brain. So to conclude, and this latest work is with Eleanor Spence, a PhD student, what I want to come back to is that by studying spatial representations, we can work out the layout of egocentric and allocentric representations of location and how they could be put together to make a neural level model of how spatial memory works. And then by understanding that, we can understand actually that this is part of a more general generative model that makes sense not just for spatial memory, but we can also think of semantic memory and consolidation, as well as imagery, all as part of the same model. And within that kind of generative model, the hippocampus is representing conceptual and sensory features, which includes place cells, and the entorhinal cortex includes these latent variables which capture the essence of scenes and allow us to generate new ones. For example, grid cells, which capture location in a way that means you can update locations, and boundary vector cells and object vector cells, which allow you to add objects and boundaries to imagined scenes. And so I'd like to thank you for listening, and also Eleanor Spence and Susanna Becker and Andrzej Bikansky and various other collaborators. Thank you very much.
Professor Neil Burgess, thank you so much, sir. We would like to keep 5 minutes for question answer session.
Yeah, Professor Burgess. It was a nice, intriguing and interesting talk. Just I'm new to this field. However, I have a question out of curiosity. So while processing this visual perception, I understand that the whole study is based on visual perception. Whether this oral or sound perception is going to complement, or is it going to create, is it going to overpower the visual perception in the process of information?
So if we're talking about these representations of location and direction in the middle of the brain, like in the hippocampus, then it seems they're multimodal. So you can use visual information or auditory information or olfaction or tactile information to tell you where you are and which way you're facing. And all of these are integrated in the hippocampus. In the sensory neocortex, then different parts of neocortex obviously represent and process the different kinds of information. And these have to be brought together somewhere to form a binding multimodal representation for memory. And that's why the hippocampus is well placed, because it gets input from all these different cortical areas and can make a high level representation which combines them all. Which one gets used most will depend on which is most useful, and visual information is usually the most useful for telling your direction and location. But if it's uncertain or it's dark, for example, then auditory information would become more important. Equally, if there's a rapid sound or something, this is very important and will cause you to orient towards it. And so different kinds of information have different uses, but need to be combined together.
Yeah, thank you, Professor.
Professor Ferguson, thank you for the lovely presentation. A lot of new things to learn. I have one basic question, just out of curiosity. That is spatial is it safe to assume that spatial memory or spatial part of the brain has to do is an evolved part of the hunting mechanism in man? Can I assume that it is directly correlated with the left brain activity or higher IQ in human beings?
Well, not specific. It's not specific to hunting. I think we see things like place cells and grid cells and head direction cells in all sorts of also in all mammals, and similar things in other species too. And this includes herbivores. So it's not specific to hunting, but it's important to remain oriented for many reasons. For example, so you don't get lost, so you know how to get back to your home, your nest, where your family are. So not specific to hunting. I think remaining well oriented in space and knowing how to find your way around is useful for many things. We know from neuropsychology, if people get damage to the left or right side of the brain, then there's some dependence on laterality in terms of which things they can't do. And it turns out that typically the left hemisphere will be dominant for memory questions, but actually the right hemisphere is more important for some spatial tasks, like if you've explored somewhere and you need to draw a map of it, or spatial visualization and recognition of faces is more dependent on the right. But if you ask anybody any questions which require verbal answers, and in general seems to be dominant, then that's more on the left. So the spatial representations that we saw, for example, the grid-like representations in the human brain, they were stronger on the right. In terms of correlations with IQ or intelligence, then different ways of measuring. There are lots of different ways of measuring IQ. Some spatial tests would correlate more with right-sided brain function, and some more verbal tests, for example, would correlate more with left-sided brain function. I don't know if that answered all of your questions.
Yes, thank you, Professor.
Good afternoon, Professor.
Hi.
Actually, when I was going through your lecture, it had a lot of parallels with your Prince of Persia game engine in computers. One of the innovations during the development of the game was the scenery that had to be projected on the screen depending on where the character is in the game. So the older games, they had multiple scenes, like Mario. So you jump from one screen to another and you navigate within the screen. Here, they actually developed what you talked. The character is present in a particular location and they are moving left or right, and the screen and the scene on the screen changes. So it was said to reduce memory storage. Means in the computer program itself, they had connected different locations to each other, and that helped them to compress the memory required for the scene generation. So is there any such thing with regards to the spatial and episodic? Spatial and episodic. Sorry, the words are not coming.
Yeah, I understand what you mean, and it's a very good question. So yes, if you test people on remembering an array of objects on a table, for example, and then you ask them questions from a different point of view when they've moved to a different viewpoint. It turns out with only one object, it doesn't really matter whether you represent the object and try and rotate the object, or whether you represent your location and imagine moving to a different viewpoint. But with lots of objects, it's much more efficient to represent all of the objects in some allocentric way, and then just imagine your viewpoint and then render what the image will look like from this viewpoint. And that's much more efficient. And that's for the same reason that the game engine, in the end, you could start off by trying to store or process all of the egocentric views that the character will have in the game. And that will work maybe with a small set of environments that you jump between, and you can see a nice background for each one. But if you want to have the character move smoothly and generate lots and lots of different views of the same scene, then it's more efficient to represent the overall layout of the environment, and then just render what the scene will look like from the point of view of the character at any given moment. And that is what we think the hippocampus is doing there. There's stored memory for the overall layout of the environment, for example, the town where you grew up. And the place cells and head direction cells can then retrieve just the relevant bits of information to create a scene from a particular location with a particular viewing direction. And that's an efficient thing for the brain to do, in the same way that it's an efficient thing for the computer program to do. And more generally, these generative networks, they make very efficient representations in the middle, so that they can generate an extremely large number of possibilities of new scenes that are consistent with what they've learned. And again, this is an efficient way of processing data. So thank you for your question.
So this helps us conserve the RAM or the ROM, like in computational terms. The current processing capacity, like with minimum processing capacity, we get a lot of information.
It's, I think, well, that's not a, so if you like, the long-term stored information, we think in long-term memory corresponding to semantic knowledge, that's, you could think of that as ROM or stored on the disk or something like that. And then the active memory, if you like, the RAM, is what you can actually have in your workspace to process at a given moment. And that's where you read out from this long-term stored information a particular scene, and be able to imagine moving your viewpoint, for example, within that scene. If you like, that's the working memory or the active memory. I guess that's more like RAM.
Thank you, Professor.
Professor, one last question. How much do you believe that this is indicating that reality could be a simulation?
Well, I mean, I don't think it's a simulation, but if we consider imagination and dreaming, then this kind of generative model shows how this kind of reality can be constructed. In fact, the generative model learns by trying to reconstruct its inputs, and it gets better and better by capturing the essence of those inputs in a better and better way. And so, I think Hello? That, can you hear me?
Yes, yes, yes.
Yeah, good. So I think that the way we generate reality internally is like a simulation, and maybe that's what's happening during dreaming and imagination, and its comparison with reality and making that better and better seems to be how we learn about the structure of the world.
Just I want to ask one question. Siberian birds, they fly about 5,000 kilometers in winter to India. Do they have that memory? What path they follow? So they fly 5,000 kilometers, but human being memory is short. They forget the rooms the birds forget. And second thing, do the plant and the tree had memory?
Sorry, what was the last question?
Plants
Oh,
have memory.
trees and plants. Okay. Well, I'm not an expert in these areas so much. In terms of migrating birds, sense of direction is very important. And I guess these head direction cells may be important for that. Studies in homing pigeons show that actually, at least in Europe, they use the smells quite a lot. They know which way the wind is bringing different smells, and that enables them to get an estimation of the large-scale layout of where these different smells might be coming from. And then when they're released somewhere far away, that gives them an idea of which direction to go in. It's also probably Earth magnetic field sensing. So we don't really know if this very long-range navigation, it could be, there are small scale grids and there are larger scale grids. Maybe there's very large scale grids and very large scale place cells. So far, they've only been recorded at several meters, not kilometers. So we don't know if you get to very large scale navigation whether different mechanisms are involved than the things we've been studying in these relatively small environments. As for memory in plants, well, so I talk mostly about episodic memory, and this is a particular kind of memory that's sort of defined as what you can consciously re-experience and reimagine, and perhaps use for planning in an explicit, conscious way. I think there are other aspects of memory. You could describe anything where your past influences your future as memory in the system overall, and plants clearly have that kind of memory in that they will grow towards the sun and so on. Perhaps if there's particular sources of water, their roots will grow in that direction. But you wouldn't call that episodic memory, but different kinds of memory you might call it.
Thank you very much, sir. I would like to extend my heartfelt gratitude to Professor Neil Burgess on behalf of Bhaktivedanta Institute for sharing your precious and valuable knowledge about spatial memory with us today. I hope everyone has gained some knowledge from this very, very comprehensive and wonderful presentation. Thank you so much again, sir, again.
Thank you. Bye-bye. Enjoy the rest of your summer school.